In the next session of topics in drug discovery, here we're going to look at physicochemical and pharmacokinetics. So the learning objectives of this session are to first define what is physicochemical in drug discovery, to understand the different physicochemical effects of drugs on their targets, and to explain the physicochemical effects in drug discovery with examples. Physicochemical is defined as the ability of a chemical compound to elicit pharmacological or therapeutic effect, and it's often related to various physical and chemical properties. So this is known as the physicochemical part of the chemical substrate. So this can be, and this is acting on a biomolecule that interacts with it. So we're going to discuss about the different aspects of physical chemical properties, including things like ionization, stability, lipophilicity, solubility, hydrogen bonding, and molecular size. So all these components actually affect the property of a drug. So being able to understand that, we can then optimize the properties of a compound to allow us then to feed this into drug formulation. So if we break it down into simple terms, so one we can see is physical properties. This is the physical property of a drug, which is responsible for its action. And two, which is the chemical properties, which is the drug which reacts with extracellular according to a simple chemical reaction like neutralization, chelation, oxidation. As discussed in the last slide, there are various physical chemical properties that contribute to drug formulation and the success of a drug. So things like solubility, which is probably the one which is at the top, partition coefficient, dissociation constant, hydrogen bonding, ionization of the drug, redox potential, which is also important, complexation, surface activity, protein binding, and isosterism. So looking at the first of these properties, solubility. Solubility plays a really important role in drug discovery, because if our drug is not soluble in solution, it's not going to work. So the solubility of substance at a given temperature is actually defined as a concentration of the dissolved solute, which is in equilibrium with the solid solute. Solubility depends on the nature of the solute or the solvent, in particular also the temperature, the pH and the pressure. So these all will contribute to the solubility. The solubility of a drug may be expressed in terms of affinity or philicity, or repulsion phobicity for either an aqueous organic solvent. So the atoms of and molecules of all organic substances are held together by these various types of bonds, as we've already discovered previously, looking at hydrogen bonds, dipole-dipole, ionic bonds. And in fact, these forces are all contributing to the solubility. So if you take a molecule which is very hydrophobic, then it's going to be fairly insoluble in a solution of water. because the solvent-solvent and solute-solute and solvent-solute interactions govern solubility. There are various techniques to enhance drugs' solubility, and such as, for example, particle size reduction, so that would help for solubility, something called nanosuspension, the use of surfactants, salt formation, and solid dispersion. Some methods to improve solubility of drugs include such as structural modifications, so altering the structure of the molecules, so looking at the chemistry, using co-solvents, so such as ethanol, sorbitol, PPG, or PEG, or employing surfactants, as we discussed, or even using complexation. The importance of solubility is that one solubility concept is important for the pharmacist because it governs the preparation of a liquid dosage form, and the drug must be in solution before it's absorbed by the body to produce a biological activity. So if it's insoluble, it's not going to work. So the drug also must be in solution form to interact with receptors. Partition coefficient, often abbreviated to P, is the ratio of concentrations of a compound in a mixture of two immiscible solvents at equilibrium. This ratio is therefore a comparison of the solubilities of a solute in these two liquids. The partition coefficient generally refers to the concentration ratio of an ionized species of compound. So the partition coefficient is one of the physicochemical parameters which is influencing the drug transport and drug distribution. So the way in which the drug reaches the site of action from the site of application. So this could be either in a tablet form or by intravenous injection. The partition coefficient is defined as this equilibrium, and we can see in this equation P, which is an ionized molecule, equals the drug, lipid, over the drug in water. For ionized so acids, bases, or salts, we use the equation of the partition coefficient of an ionized molecule equals the drug in lipid over 1 minus A. A is the degree of ionization in an aqueous solution times the drug in water. So the partition coefficient affects the drug transfer characteristics, the contribution of each functional group or structural arrangement can help to determine the lipophilic or hydrophilic character of the drug. This is widely used in quantitative structure, activity, relationship. So in pharmacokinetics, this is where what the body does to a drug. The distribution coefficient often has a strong influence on something called the ADME properties, which we've discussed before, the drug. So the hydrophobicity of a compound can be measured by its distribution coefficient, and this is a major determinant of how drug-like it is. So to be more specific, for a drug to be orally absorbed, it must pass through some lipid bilayers in the intestinal epithelium. And this is known as something called transcellular transport. The efficient transport of a drug must be hydrophobic enough to partition into the lipid bilayer, but not so hydrophobic to affect its solubility, but also that once it's in the bilayer, it will not partition out again. Also, hydrophobicity plays a major role in determining the drugs are distributed within the body after absorption. And the challenge is actually if we need to target the brain, we need to design the drug in a certain way that it can pass through the blood-brain barrier. And we can see in the figure here, this shows us the drug permeability in brain capillaries, which is on the y-axis, as a function of the partition coefficient on the x-axis. And we can see some examples of different drugs, for example, actually hormones, which obviously can pass through the blood-brain barrier. We can see that the different partition coefficients. So there's a number of factors that are affecting the partition coefficient. So we've got pH, co-solvents, surfactants such as PEG, complexation. So the partition coefficients are difficult to measure in a living system. There are many factors going on. So they're actually usually determined in vitro using 1-octanol as a lipid phase and phosphate buffer at a physiological pH of 7.4 as the aqueous phase. So 1-octanol as a lipid phase because it has a polar and non-polar region, which I've written here PO over W, which means the partition coefficient N-octanol over water. That's easy to measure. Also, PO is often correlated with many biological properties, and it can be predicted using computational mode. The partition coefficient P is dimensionless, and it's logarithmic. So logP is widely used as a measure of lipophilicity. The logP is measured by the following methods. So 1, a shaking flask method, or 2, chromatographic methods such as HPLC. Phenobarbital has a high lipid water partition coefficient of about 5.9, whilst thiopentone has a chloroform water partition coefficient of about 100. So this is highly soluble in lipid. And it's a reason why it can pass through the blood-brain barrier, because it's a lipophilic molecule. And hence thiopentone is used as an ultra-short-acting barbiturate. So what else does logP affect? So with increasing logP, we can get increasing binding to the enzyme receptors, a decrease in aqueous solubility, an increase in the binding of P450 metabolizing enzymes, an increase in absorbance through the membrane, and the binding to blood tissue proteins. So the importance of partition coefficient. It's generally used in combination with pKa to predict the distribution of a drug in a biological system. The factors such as absorption, excretion, penetration of the central nervous system, this may be related to the logP value of a drug. And so the drug needs to be designed with the lowest possible. logP may also contribute to reducing toxicity and also non-specific binding and bioavailability. Hydrogen bonding. So hydrogen bonding is a special dipole-dipole interaction between the hydrogen bond in a polar bond, such as nitrogen-hydrogen, oxygen-hydrogen, fluorine-hydrogen, and also electronegative atom, oxygen-nitrogen or fluorine atom. Dipoles result from unequal sharing of the electrons between the atoms within the covalent bond, and these are weak bonds, so denoted as dotted lines. The compounds are capable of forming hydrogen bonding, is only soluble in water, so hydrogen bonding can be classified into 2 types. 1, intermolecular, 2, intramolecular. Intermolecular hydrogen bonding, so this occurs between 2 or more than 2 molecules of the same or different compound, due to an increase the boiling point of compound and the increase in molecular weight of compound, hence more energy is required to dissociate the molecule for vaporization. Intramolecular bonding, so hydrogen bonding occurs with 2 atoms of the same molecule. So this type of bonding is known as chelation and it frequently occurs in organic compounds. Sometimes the hydrogen bond develops 6 or 5 member rings, and that's due to a decrease in the boiling point. The first example, antipyrine. This is an analgesic or non-steroidal anti-inflammatory drug, or known as NSAID. It's used to relieve pain and fever, and is often used in testing the effects of other drugs or diseases on drug metabolizing enzymes in the liver. As you can see in the top, this is the active form, producing the analgesic activity, whilst the 1-phenol-free methyl 5-pyroxoline is an inactive form. The other example is salicylic acid, known as O-hydroxybenzoic acid. This has antibacterial activity and is often used in medication to remove the outer layer of skin, and is used to treat things like warts, acne, wingworm, dandruff, etc. As you can see, another form, which is called the parameta hydroxybenzoic acid, these are inactive. So hydrogen bonding can affect many physical properties of a molecule, so including changing the boiling melting points, altering its water solubility, the strength of an acid, the spectroscopic properties, even things like surface tension and viscosity of a liquid, the biological products, and drug receptor interaction. Chelation or complexation. Chelation is a process of reversible binding, so complexation, of a ligand. The chelant or chelator or chelating agent or sequestrating agents, they act. The term is generally reserved for complexes in which a metal ion is bound to 2 or more atoms of the chelating agent, although the bonds may be any combination of coordination or ionic bonds. So a complex drug molecule can't cross the natural membrane barrier. They render the drug biologically ineffective. So the rate of absorption is proportional to the concentration of the free drug molecule. So this is known as the diffusion of the drug. Due to the reversibility of the complexation, the equilibrium between free drug and drug complex. Complexation reduces the rate of absorption of a drug, but not affect the availability of the drug. Regarding the importance of chelators in medicine, for example, as an antidote for metal poisoning, so 1-dimercaprol is a chelating reagent, is actually used as a mainstay of chelation therapy for lead or arsenic poisoning, and it remains as an essential drug. So it's actually an antidote to a chemical weapon called logP. Penicillamine is a monophile chelating agent, which is used in veterinary medicine for the treatment of copper storage hepatopathy, also known as Bedlington Terriers. Also for lead toxicity, it's being used in the management of rheumatoid arthritis in humans. 8-hydroxyquinoline and its analogues can act as antibacterial, antifungal agents by complexing with iron or copper. But there's undesirable side effects caused by drugs which chelate metals. A side effect of hydrolazine, which is an antihypertensive agent, is the formation of the causing agent of anemia, and is due to the chelation of the drug with iron. Phenobarbital forms a non-absorbable complex with polyethylene glycol 4000, and calcium with EDTA form a complex which increase the permeability of the membrane. Chelation therapy could be an alternative treatment that could be used to remove heavy metals or toxins from the body by using chemicals. In fact, it's been used for a long time to treat kind of lead and mercury poisoning, as we've kind of discussed before.
But now actually many people believe that it can be of benefit to people who are suffering from like heart disease, so maybe as a preventative measure, and other health problems. So the example I've got on the slide is looking at kind of using chelation therapy. So chelation therapy can be essentially conducted through intravenous drip. And this allows the chemical substance then to enter the blood circulation. And then the chemical then can move around in the bloodstream and then bind to any kind of toxic metals or minerals found in the body and kind of help to remove them. EDTA can actually form, it's just a kind of form of amino acid that kind of commonly used to remove substances, so such as lead, copper, calcium. And these are kind of deposits which we find around the body. So this type of therapy could be used to treat atherosclerosis. So atherosclerosis is the kind of chronic disease where you get plaques building up on the walls of the arteries. And these plaques are then caused by the deposits of those lipids and cholesterol and calcium. And the plaques can essentially cause many problems and stop kind of the blood flow and also cause blood clotting and weakening of the arterial wall. So chelation therapy could essentially help to remove calcium deposits and also then hopefully to kind of restore the blood flow into those arteries.
When we look at ionization of a drug, most drugs are either weak acids or bases, and they can exist in either ionized or unionized form. So ionization is the protonation or deprotonation, which results in a charged molecule. The ionization of a drug depends on its pKa and also its pH. And the rate of drug absorption is directly proportional to the concentration of the drug at its soluble form, but not the concentration of the drug at the absorption site. Ionization form imparts a good water solubility of the drug, which is required for the binding of the drug to its receptor for its interaction. Unionized form helps the drug then to cross the cell membrane. So there's a benefits of having ionization and unionization. For example, barbituric acid, which is an inactive because it is a strong acid, while 5,5-disubstituted barbituric acid is a CNS, has CNS action because it's a weak acid. According to the Henderson-Hasselbalch equation for acids, when an acid or a base is 50% ionized, so the pH equals the pKa, for example, a solution of weak acid aspirin in the stomach at a pH of 1, so in a very acidic environment, will be readily absorbed because it's in its unionized form at 99%. Phenytoin injection is used to treat for treatment of seizures occurring during or following neurosurgery or after severe head injury. Phenytoin injection has to be adjusted to a pH of 12 with sodium hydroxide to obtain a 99.98% of drug in the ionized form. Another example is tropicamide eye drops, which is an anticholinergic drug, has a pKa of 5.2, and the drug has to be buffered to pH 4 to obtain more than 90% ionization. A weak acid, at an acid pH, more lipid soluble because it's uncharged. The uncharged form is more readily passes through the biological membranes. A weak base, at an alkaline pH, more lipid soluble because it's uncharged. The uncharged form more readily passes through the biological membranes. Redox potential. So the oxidation reduction potential may be defined as a quantitative expression of the tendency that a compound has to give or receive electrons. So there's a correlation between redox potential and biological activity that can be drawn for a compound of a very similar structure and property. So the redox potential of a system may be calculated from the following equation. E equals E0 plus 0.592 over n the log, which is the concentration of a reductant, over divided by the concentration of oxidant. Riboflavin analogues, so the biological activity of riboflavin is due to a redox potential of minus 0.18 volts, whilst dichlororiboflavin has a redox potential of minus 0.095 volts. The optimum bacteriostatic activity in quinones is actually associated with a redox potential at a positive 0.03 volts when tested against Staphylococcus aureus. Surface activity. So a surfactant is defined as a material that can reduce surface tension of water at very low concentration. So surface active agents affect the drug absorption. That depends on 1, the chemical nature of the surfactant, 2, its concentration, 3, its effect on the biological membrane and the micelle formation. In low concentration of surfactant, there's an enhanced rate of absorption because the amplifiers reduce the surface tension and there's better absorption. In a higher concentration of surfactant, there is a reduced rate of absorption. Applications. So anthelmintic activity of hexylresorphenol. These are essentially drugs that either kill or expel infesting helminths, and the gastrointestinal tract is the abode of many helminths. Although they can also live in tissues, and the larvae can migrate into tissues. Also, it's been used in bactericidal activity of cationic quaternary ammonium compound, bactericidal activity of aliphatic alcohols, disinfection action of phenol and creosol, biosalt solutions of approximately physiological concentration greatly enhance the dissolution rate of poorly water-soluble drugs like grasulfulvin, hexatrol by micellar solubilization effect. Protein binding. So the residual binding of a protein with a non-specific and non-functional site on a body of a protein without showing any biological effect is called protein binding. So protein plus drug equals protein-drug complex. Depending on whether the drug is a weak or strong acid or base or is neutral, it can bind to a single blood protein to multiple proteins, such as serine albumin, acids, glycoprotein, or lipoproteins. And the most significant protein involved in the binding of the drug is albumin, which comprises more than half of the blood proteins. Protein binding values are normally given as a percentage of total plasma concentration of drug that is bound to a plasma protein. Free drug plus free protein is in an equilibrium of drug-protein complex. Total plasma concentration, dt, equals dF plus dP. Sterochemistry. Sterochemistry involves the study of three-dimensional nature of molecules. It's the study of the chiral molecules. It plays a role in the pharmacological properties because of two things. One, any change in stereospecificity of a drug will affect its pharmacological activity. Two, the isomeric pairs have different physical properties, logP, pKa, and thus differ in pharmacological activity. So the isomer which have the same bond connectivity but different arrangement of groups or atoms in space are termed stereoisomer. Conformational isomers, so different arrangements of atoms can be converted into one another by the rotation of a single bond, which are called conformations. The rotation of about a bond allows interconversion of conformers. So the rotation of about a single bond of butane to interconvert one conformation to another. The gauche conformation on the right is a conformer, whilst the eclipsed conformation on the left is a transition state between the conformers. This slide shows the classical example of acetylcholine, which can actually exist in different conformations. So it can exist as a trans-conformer, a Gauss conformer, a trans-conformer staggered or a Gauss conformer, or an eclipsed form. Optical isomers, so essentially these are like mirror images of each other. Sterochemistry, enantiomers, symmetry of chirality are important concepts in therapeutic and toxic effect of drugs. The chiral compound containing one asymmetric center has two enantiomers. Although each enantiomer has an identical chemical and physical property, they may have different physiological activity, like for example the interaction with receptor, or metabolism, or protein binding. The optical isomers in a biological action is due to one isomer being able to achieve a 3-point attachment with the receptor molecule, while its enantiomer would only be able to achieve a 2-point attachment with the same molecule, which would have then a low binding efficiency. The example above shows lactic acid. So on the left is the S-positive lactic acid, on the right is the minus lactic acid. They are non-superimposable mirror images of each other. Ephedrine is a medication or stimulant. It's often used to prevent low blood pressure during spinal anesthesia. It is also used to treat asthma or narcolepsy or obesity, but is not the preferred treatment. It has an unclear benefit in the nasal congestion. Ephedrine exhibits optical isomerism and has two chiral centers, giving rise to these stereoisomers. So, by convention, the pair of the enantiomers with the stereochemistry are designated ephedrine, while the pair of enantiomers with stereochemistry is called pseudo-ephedrine. Ephedrine is a substituted amphetamine and has a structural methamphetamine analog, and this differs from the methamphetamine only by the presence of a hydroxyl group. Geometric isomerism is represented by cis or trans isomerism, resulting from restriction or restricted rotation due to the carbon-carbon double bond or in a rigid ring system. Diethyl silbestrol, or DES, is a non-steroidal estrogen medication, which is rarely used, but in the past is being used widely for a variety of indications, including pregnancy support for women with history of recurring miscarriage or hormone therapy for menopausal symptoms. It's also being used for the treatment of prostate cancer, but today now the treatment of prostate cancer, or less commonly breast cancer. The chemical structures of estradiol and DES note there is actually a preservation of the two hydroxyl groups in DES and similar distance between the relative estradiol, which is notable when it's considered that DES was first discovered serendipitously. Longmore introduced the term isosterism in 1919, and which he postulated that two molecules or molecular fragments containing an identical number and arrangement of electrons should have similar properties and termed as isosteres. Isosteres should be isoelectric, so they possess the same total charge. Biosterism is defined as compounds or groups that possess near or equal molecular shapes or volumes, approximately the same distribution of electron, which exhibit similar physical properties. They are classed into two types: classical biosteres and non-classical biosteres. Classical isosteres, they have similarities of shape and electronic configuration of atoms and groups and molecules which they replace. The classical biosteres may be univalent atoms and groups, such as chlorine, bromide, etc., or bivalent groups. Classical bioisosteres have a number of applications in drug design. For example, the replacement of the hydrogen atom with a fluorine atom at a site of metabolic oxidation in a drug candidate may prevent such metabolism from taking place. Because the fluorine atom is similar in size to the hydrogen atom, the overall topology of the molecule is not significantly affected, actually leaving the desired biological activity unaffected. However, with a blocked pathway metabolism, the drug candidate may have a longer half-life. Procadamide, it's an amide, is actually a classical bioisostere because it has the valence electron structure of a substituted oxygen atom, has a longer duration of action than procaine, and is an ester because of its isosteric replacement of the ester oxygen with a nitrogen atom.
Next, if we consider some examples of isosteres. So, for example, acetopram, which is an antidepressant known as a kind of selective serotonin reuptake inhibitor. If you take this hydrogen, okay, so this hydrogen, it can be replaced by fluorine, okay, and the problem with this hydrogen is that it can undergo phase 1 oxidation, and this actually reduces the half-life of the molecule. So by replacing it with a fluorine, this would increase the half-life. Also, the change in the molecule doesn't really change the kind of bulk, kind of steric bulk of the molecule. And in fact, this atomic radius of the hydrogen is around about 1.2 angstroms, whilst fluorine is about 1.35. And so by replacing this kind of small group, we don't affect the potency. In our second example, if we look at tolbutamide is a type 2 diabetes drug. If we look at the methyl group, okay, so the methyl group, which can also undergo phase 1 oxidation, can actually be replaced by chlorine. And by replacing the methyl group by the chlorine, we can actually increase the half-life of this drug. So from 7 hours for tolbutamide to 36 hours for chloropropamide. So in our next example are chalcones biostere. So chalcone essentially is an aromatic ketone. So here we've got the aromatic ketone part, which has this enone, which kind of forms a central core. And by changing some of the kind of side chains, so here we've got the 2 bromides on this aromatic ketone, and here we have an OH group and a double bonded oxygen. So by changing these side chains on these different molecules, we can alter the minimum inhibitory concentration. So here this one has MIC of 2 micromolar, whereas this one has 20 micromolar. The difference being you have a chlorine here instead of bromide, and also a chlorine here. Also, we lose this double bonded oxygen, just keep the hydroxyl group. The difference between these two molecules are that this one has contributes to kind of high solubility, but is a bacteriostatic, meaning that it kind of stops the bacteria from growing but doesn't kill them. Whilst this one has low solubility but is a bactericide, meaning that it kills the bacteria. In our third example here, this one has a similar structure to the first one. It's got the OH group and this double bond oxygen, but instead over here on this aromatic ketone, it has 3 fluorines, and over here as well 3 fluorines. And this, again, it still keeps its MIC, so the minimum inhibitory concentration of 2 micromolar. It also has high solubility, and it's also a bacteriostatic.
For non-classical biostere, they don't obey any steric or electronic definition of a classical biostere. So they are non-functional groups with dissimilar valence electron configuration. They have specific characteristics, so electronic properties, physicochemical property of the molecule, spatial arrangement, and functional moiety for biological activity. Examples of non-classical biostere are alloxane. Alloxane is an inhibitor of xanthine oxidase. It's also an isostere of xanthine, which is a normal substrate for this enzyme. The other one is silafluorofen, which is an organosilicon analog of the insecticide Atofen prox, wherein the carbon center, as you can see in the figure, has been replaced by isosteric silicon, and in addition, one hydrogen atom is replaced by an isosteric fluorine atom.
So, in summary to this session, we have considered the definition of physicochemical, and that's the ability of a chemical compound to elicit a pharmacological therapeutic effect in related to the influence of various physical and chemical properties of the chemical substance on the biomolecule that it interacts with. So the physical chemical property of a drug is governed by 1, the physical properties of the drug, which is responsible for its action, and 2, the chemical properties of the drug, which kind of reacts extracellularly according to simple chemical reactions like neutralization, chelation, oxidation. So there are various physical chemical properties that govern the efficacy of a drug, and we have considered, for example, solubility, partition coefficient, dissociation constant. The hydrogen bonding of a drug can affect the potency of the drug, and also the half-life, the ionization of the drug, the redox potential, so the ability to kind of cross the membranes, the complexation of the drug with its target, the surface activity, things like protein binding, and also isosterism. The last slide is kind of some further reading to take a look at, and the first is a book chapter from Florence and Atwood. So this is Physical Chemical Principles of Pharmacy, Manufacture, Formulation and Clinical Use. Now there's a number of, to have a look through this book because there's a number of interesting chapters in this book. Also there's a couple of papers to have a look at. This one from Cunningham Rooker in 2017, looking at octanol water partition coefficient measurement by simple proton MMR method. And the next one is from Bali 2020. It's chemical reactivity and uses of 1-phenol-3-methyl-5 pravassolone, also known as endovorine. And the next one is looking at, this is from Suhail et al. In 2019, looking at surfactants and their role in pharmaceutical product development. And this is a review. In the next one, this is also a review looking at structural simplification from Wang et al. In 2019, an efficient strategy in lead optimization. And finally, in the last review, which is quite a nice review, is from Bonali. This is using physicochemical measurements to influence better compound design